So we are here, we are at our final step of respiration. We're in the step of oxidative phosphorylation. Now, I understand that this is a long word, and I understand that people may be put off because it is a long word, but it's actually very, very simple, as most things with biology and chemistry are, with a little bit of understanding. So, oxidative. This just means that it's in the presence of oxygen, really. And then we have the word phosphorylation. To phosphorylate something means adding a phosphate. So hopefully we can imagine what it is we may be phosphorylating. Yeah. The whole point in respiration is to produce adenosine triphosphate, which is how all of our energy is contained and transported and used is in the form of ATP. To produce ATP from ADP, adenosine diphosphate, we phosphorylate it. We add a phosphate group to make ATP. So all the name means is phosphorylation of something, air, in the presence of oxygen. I'll explain that later. Also, before I get started, I do want to refer to the term electron transport chain, otherwise known as the ETC, because there are going to be some A-level students watching this thinking, I thought it was called the electron transport chain. I know that a lot of students will think this because that's what I was always told, and that's what I always thought, but it's not actually what it is. The electron transport chain is part of oxidative phosphorylation. It is not the name of the step. So, getting onto it, this all takes place in a mitochondria. They are an organelle that exist inside the cell, and they are the step for the link reaction, Krebs cycle, and this oxidative phosphorylation. We know already that glycolysis doesn't take place in the mitochondria because it has to split the glucose to get it to fit inside. That's the whole point of it. So, we have this mitochondria. The inner membrane, as it has two, is folded into cristae. And cristae is where all of this funk jam magic is taking place. So, let's draw our cristae here. This inner membrane is studded with proteins. And these go down the length of the cristae. Know that NADH and FADH2 that I've been cracking on about? Well, this is where it's going to be put into use, because they are proton carriers, but they're also kind of electron carriers as well, because they will split, I'm just going to draw it upside down to be difficult, into FAD and NAD, and they will then go back and return to the previous steps to be reused. We only have so much of it, we need to reuse it, and if we don't do this, then every other step can't take place because there's nothing there to be reduced. It will also produce electrons as well as protons, and each one will go their own way, which I'll explain now. So our electrons will first go into this protein here. As it's the addition of an electron, the protein has been reduced because reduction is gain of electrons. So that's reduced protein. This electron will then leave, causing it to be oxidized because oxidation is loss of electrons. That will then reduce the next. It will then leave that one, causing it to be oxidized, and reduce the next. This produces a series of reduction and oxidation summarized as redox reactions. Now, this redox produces energy, not in the form of ATP, but it still produces energy. We can actually use this energy to actively transport those hydrogen ions produced up here into the membrane, because the membrane is really highly concentrated with protons already. That means there's going to be a lower count out here than there is inside the membrane, thus necessitating active transport. And if we didn't have the electron doing these redox reactions, we wouldn't have the energy to be able to move them in. Once inside the membrane, protons can't leave because the membrane's pretty tight. This means they can only leave through a specific protein channel, and this channel is called ATP synthase. I'll give you 3 guesses as to why it's called that. So the protons will leave through ATP synthase, and they will go to the matrix of the mitochondria. They will leave the intermembrane space. They do this by diffusion because obviously there's a lower concentration out here than there is in here, so they're just moving down their concentration gradient. The cool part is as it leaves the ATP synthase, it causes the joining of ATP plus P to produce ATP. And this is how we produce energy. This is how we get energy from our food. After all of that, we've made these proton carriers. The protons go into the membrane, they leave by diffusion, and as they leave, the energy released is harnessed, and it is used to join ADP and P to make ATP. We now have our energy. This is fantastic. The only problem is we now have an electron stuck here and a proton stuck here. Now, if the protons kept building up, it would ruin the concentration gradient and they wouldn't leave through diffusion because they'd be pretty much in equilibrium. And we couldn't have electrons moving down because these proteins would be constantly reduced. This is where oxygen comes in. So we have O2 coming in, that will be split into 2 molecules of oxygen, and we have our oxygen here. This electron can now leave that and enter the oxygen, which is why oxygen is referred to as the final electron acceptor. That is exactly what it does. It is the final thing to accept this electron, and it is then reduced. It will then join with this proton as well as one more to form H2O. And this is why we expire water and we inspire oxygen. That is going to be exhaled. And we have gotten rid of these protons to prevent the buildup out here, to keep the concentration gradient, and this can continue to happen to produce the energy to put the protons in. And also, if we didn't have anything to accept this electron, it would stop this happening, which means we wouldn't have FAD and NAD back, which means that glycolysis couldn't happen. It means that when the acetyl coenzyme A is formed, we couldn't produce the NADH, so that would stop that reaction. It would also stop the Krebs cycle because FAD and NAD are also produced in that. So we need to be able to maintain all systems, and we do this by forming water. So, fantastic. That is every single stage of respiration gone through now. If you found this useful, make sure to leave a thumbs up, subscribe, share the video if you like, just whatever. It will really help the site out. I really appreciate it, and I hope that it helped.